Okay, so this is our second talk of the Worms of the World event, and this is the phylogenetic tools to dig out the evolutionary history of earthworms with Dr. Daniel Marchán. Daniel is an earthworm researcher integrating the fields of molecular phylogenetics, morphology, and ecological methods, such as ecological niche modeling, and he's going to talk us through how his research is helping us better understand earthworms, particularly, I think, across Europe. So I'm going to hand over to Daniel now, and off you go, Daniel.
Thank you, Karen. Well, hi, everyone. As Karen said, I'm going to tell you how those phylogenetic tools help us dig out the evolutionary history of earthworms. Well, here you can see how you literally dig them out, but usually that's not enough to understand them. So we go into those other tools. I'm going to turn off my video because it's doing really weird things, okay? I think that's better. I'm not possessed or anything. Not for the moment. The slides are not working, I think. Yeah, now. Okay, so a traditional taxonomy of earthworms have some limitations related to their morphology. First of all, they have a really limited number of morphological characters. That's because they are soft-bodied, so they don't have any skeleton or similar structures that you can use to identify them, and their body plan is very conserved. Their body plan is pretty much the same across the different families. They also have a lot of simplesiomorphic characters, which means they are inherited from a distant common ancestor. So they don't tell us a lot about their phylogenetic relationships. Also, many of their characters are homoplasious. This means that they evolve convergently. Again, they don't tell us a lot about phylogenetic relationships. That has led to an unstable taxonomy of earthworms. As you can see here, in the 20th century, there was a quick succession of different genera systems, and this makes it very difficult to classify them. That's why we have used molecular phylogenetic tools, which have allowed us to advance a lot on earthworm systematics, but those techniques have advantages and also pitfalls. Here you can see a graph which summarizes most of those techniques, and the main idea is that, as you can see in those arrows, when you go up there, you increase the coverage of populations or species, but you lose the representation of the genome. So you get more species or more population, but less a lower percentage of the genome represent. And the opposite, if you go down there, you get a larger representation of the genome, but you get less coverage of populations or species. One of the first techniques, molecular techniques, which was applied to earthworms was a DNA barcoding, which consists on the sequencing of the cytochrome C oxidase subunit 1 gene. This technique has allowed the discrimination of closely related species and genetic lineages within a single species. It has also allowed to discover species-level cryptic lineages across different earthworm families, and this phenomenon appeared to be widespread. And also, it has allowed to perform phylogeographic analysis. Those phylogeographic analyses require a wide representation of individuals from different populations and across their whole range. Another method, a bit further down in the scale, is the multi-gene or multi-locus phylogenetic analysis, which consists on sequencing several individual mitochondrial or nuclear molecular markers, and this allows this phylogenetic analysis allows to recover closely related species within monophyletic clades. This usually means genera. So it allows to place an earthworm in its corresponding genera And sometimes it even allows to discover relationships between the genera. But in some cases, such as ancient divergence or rapid radiations, this approach lacks power. Then the ideal approach would be whole genome sequencing. Whole genome sequencing would provide information about all the genome. So in that scale, it would be there, but it's still unaffordable for most phylogenetic applications because you can afford to have like one representative of one species, but not all the species of one family, for example, but you can have several representatives of several species within a family. So for most applications, it is not useful. An alternative is transcriptomics, which has already been applied to earthworms. This consists on taking all the genes which are expressing in a certain moment and sequencing them. So it provides quite a lot of information about the genome. And this method allows phylogenetic resolution at family level and even above family level. But the pitfalls are several. First of all, it requires very complex bioinformatic pipelines, and also sometimes genes evolving at different rates can provide different topologies. So you don't really know which one of those topologies is the good one. And it also requires freshly preserved specimens, and the preservation and handling of RNA is very difficult. So it's not an ideal method. An alternative is target enrichment methods, which are mainly UCE and AHE, which need the existence of a previous genome for a closely related species. And with that genome, you can select the loci that you want to, the genes that you want to amplify, and design probes. And this is a method which is efficient and inexpensive, and that provides hundreds, like for example, 600 orthologs or independent genes, as you can see here. And for example, AHE provide phylogenetic signal both at deep, it means like between families, and shallow, like between populations scales. So it's pretty much a very versatile tool. Then there are the reduced representation techniques such as RADSEQ or GBS, which rely on restriction enzymes, which cut the genome in several places, and then those fragments are sorted by size, and all of them are sequences. So again, it provides information about quite a lot of the genome. It allows to obtain thousands of single nucleotide polymorphisms, which are suitable for population genetics, for selection signature analysis, and cryptic lineage delimitation. So a very useful method. Well, so now I'm going to tell you the advances that we have made on the systematics of the European earthworms, mostly, and what it teaches us about their historical biogeography and morphological evolution. For example, for the family Lumbricidae, which thankfully Sam in his awesome talk has already introduced them. For them, the starting point was the multi-gene phylogenetic analysis of Jorge Dominguez and collaborators, which highlighted some of the most obvious problems of the systematics of Lumbricidae, which is rather troublesome. For example, several Lumbricid genera were found to be paraphyletic or polyphyletic. This basically means that those genera don't contain all the descendants of a species, or that they contain a species belonging to unrelated clades. So some of those genera were Aporectodea, Alolobophora, Dendrobaena, or Helodrynx. Others, on the other hand, others appear to be monophyletic. For example, Eisenia, Sclerotheca, or Lumbricus. And this analysis lacked a strong representation of rare or endemic genera, and those genera has been slowly added to the phylogeny of Lumbricidae. For example, again, Dominguez and collaborators in 2018 discovered 2 new genera from northwestern Iberian Peninsula, mostly from Galicia and Leon, which are called Galiciadrilus and Compostelandrilus. Those genera, surprisingly, are closely related to the Balearic Postandrilus, and they have an interesting character. In Postandrilus, all the species present backward displaced male pores. They should be in segment 15, but they are usually found on segment 30 or 40, like very strongly displaced backward. But in Galiciadrilus and Compostelandrilus, we find this character in some species, but not in others. With that indicate us is that the backwardly displaced male pore is a homoplacious character state, and it doesn't give us information about phylogenetic relationships. Those genera that I have mentioned before are related to the French Critodrilus, which are found there, and the newly described Central European genus Bindoboscolex. So these tell us some things about the early historical biogeography of Lumbricidae. There was an early branching clade here in the tree, which occupied a wide area going from Galicia to Hungary, and this lineage was later fragmented into relict, geographically restricted clades, probably because of competition with other more modern earthworms, but also because of climate change. So one of those troublesome genera that I mentioned, Alolobophora, it's currently the second most diverse genus of Lumbricidae with 69 species, but we know that it's a taxonomic wastebasket. This means that many unrelated species have been put into this genus, mostly because the diagnosis of the genus or the characters that define the genus are very lax. So everything goes. Thankfully, by using molecular phylogenetics, we have managed to divide Alolobophora into Gattasona, Cernosvitobia, Pannoniana, Bosgesia, Lukesia, and Alolobophora sensu stricto, or like the real thing. For example, the type species Alolobophora chlorotica, we can see here that it forms a monophyletic clade with the green pigmented earthworms Alolobophora dubiosa and Alolobophora molleri and moebi, but also with the French species Alolobophora icterica and Alolobophora burgundiae. Then an unrelated clade contains Carpato-Balkanic species which were formerly assigned to Alolobophora, such as Alolobophora mehadiensis or Alolobophora robusta. These species form a monophyletic clade with the species called Cernosvitobia, so this led us to redefine Cernosvitobia to contain these species which are no longer Alolobophora. And again, in 2021, we found that Elodrilus cortesi, Elodrilus musicus, and Alolobophora chetophora, plus several subspecies which were elevated to species, they form a monophyletic clade, which is actually the genus Gattasona, and it's a genus related restricted to France. We also found more recently in this year that Alolobophora tiginosa and Alolobophora thixi are actually two separate genus, Lukesia tiginosa and Bosgesia thixi, which are French relic genera. They are only in France and restricted to very small areas. Plus, we found that Pannoniana is also not related to Alolobophora. It contains perialpine and Transaegean species. So this division has resulted in 6 different genera which have a more coherent biogeography. Previously, all those species were in a single genus, which had a really wide range. But now we can see that those genera have like more coherent distributions, more restricted, and only Alolobophora sensu stricto appears to have like a wide distribution, but it makes a lot more sense. And this happens pretty much with any family of earthworms that we study. that large genera get split into small genera, which are restricted geographically. Aporectodea is another very troublesome genus, and it's one of the most, it contains the most widespread and common lumbricids, which you may be familiar with, such as Aporectodea caliginosa, Aporectodea trapezoides, or Aporectodea rosea. When we have applied molecular phylogenetics, we have found that Aporectodea trapezoides, which is the type species of the genus, Aporectodea caliginosa 2, and other species which were placed by Marcel Boucher into Nicodrillus, they form a well-supported clade, seen here, which is completely unrelated to Aporectodea rosea or Aporectodea georgii, which have been placed in the genus Coinodrillus, but also other species which were placed in Aporectodea, such as Limicola, which as far as I know is present in the UK, or Aporectodea pseudoantipi. So all those Aporectodea species which are not closely related to Aporectodea trapezoides, all those can't belong to Aporectodea, and they must be placed within newly defined genera. We are currently working on that. We also know, thanks to molecular phylogenetics, that there are cryptic species within Aporectodea sensulato, in Aporectodea caliginosa, in Aporectodea trapezoides, and in Aporectodea rosea. Those deeply divergent lineages deserve to be described as different species, and we must do so, because when you do ecological studies and you say, okay, I found Aporectodea caliginosa, but actually you found 3 different species, you are getting wrong results. Or if you do a test with a chemical and you say, oh, Aporectodea caliginosa appears to be fine, but you use representatives of 3 different species without knowing it, that's a problem. And the main problem is that describing the species which are virtually identical in morphology is difficult. And it's also difficult to assign the holotypes to one of those lineages. So we are currently working on it. Well, now I'm going to talk about the family Hormogasturidae. Again, Sam introduced them. They are comparatively small, but well-represented family in the Western Mediterranean, and they are ideal because they have few species for integrative systematics. Which is integrative systematics? Well, it's combining morphological characters and molecular phylogenetics. Well, transcriptomics, which I talked to you about, allowed us to place the very strange Aeolus and Hemigastrodrillus, which Sam mentioned, both are very strange earthworms, as early branching clades of the family, like the most basal members of Hormogasturidae. Also, the integration of morphological and molecular morphological characters and molecular markers allowed to revise the genus system of Hormogasturidae. The previous genus Hormogaster was divided into Hormogaster sensu stricto, Busheona, there, Norana, there, and Carpetani. And also, we found that the species Hormogaster preciosa should be limited to the specimens in Sardinia, and that the French and Spanish populations called Hormogaster preciosa were actually different species, and they must be described as them. And currently we have advanced a lot on that front. Also, we found that the genus Carpetania, which was formerly described as the complex Hormogaster release, is an ideal model for studying the phylogeography of cryptic lineages, and also the evolutionary process behind their diversification. For that, we used GBS, which I mentioned before, and this GBS allowed us to delimit 3 large species within this genus and to describe them through integrative methods. Another interesting approach to Hormogasturidae was the application of the phylogenetic comparative method, which has been used extensively in vertebrates, but rarely in invertebrates, to study macroevolutionary patterns. For example, the origin and radiation of a key evolutionary innovation, which is the multilamellar typhlozoal. You can see here the typhlozoal, it usually looks like a simple lamella, but here is formed by 3 lamella. And in the evolution and radiation of the family, it gets more and more complex. And here it appears like the pages of a book. It can reach like 21 lamella. So we wanted to see to what in what degree this character had affected the evolution of the family. And what we saw is that this character appears to be linked to the appearance of large body weight. When we have larger species like this one, which is kind of giant, we find that they have a large amount of lamella, but when we have a small species, they have very few lamella. So it appears as if this character allowed the family to radiate and to achieve those giant sizes. Well, and these molecular phylogenetic approaches are a good give us a good possibility to understand the functional trait evolution in Lumbricoidea. Lumbricoidea here would include Ormogasturidae and Lumbricidae, and more precisely to study the evolution of large body size. And you have heard Sam mention large body size several times, and well, he also mentioned the Sceroteca, which is a very interesting genus restricted to southern France, northern Spain, and Italy, and their species have very narrow ranges. Some species, like this one, are mid-sized Indogic and resembling a Proctodea caliginosa a lot, but most species display large or even gigantic sizes, like you see here, which I define as having a length over 30 centimeters, diameters over 1 centimeter, and weights over 20 grams. And those earthworms have usually anethic lifestyles. I hope you are familiar with the word anethic. If not, I will clarify it in the questions. And here when we build the molecular phylogenetics of the genus Sceroteca, and we plot into it its size and pigmentation, we find three large clades, which make sense biogeographically: Corsica, Sardinia, and Provence, Occitanie, Provence, and Catalonia, and southwestern France and northeastern Spain. And we see that in each of those clades, except on Corsica, which are mostly like miniaturized versions, in the other two, we find that giant anethic forms have evolved independently like several times, which is very interesting. And we have Octodrilus, which is surprisingly, it's a genus which is very convergent. It has similarly high diversity, a similar size range with a small and giant species, and also anethic lifestyles, but they inhabit mainly Italy and the Balkans. Plus, some genera of the family Ormogasturidae have similar biogeography and morphological radiation to the previous two. Their ancestral state appears to be small or mid-sized Indogic, but they have evolved independently the anethic lifestyle and large to giant species, as you can see here. So, interestingly, Sceroteca, Octodrilus, and the large Ormogasturidae show non-overlapping ranges. Octodrilus here, with some invasive here. This is Sceroteca, and this is the large Ormogasturidae. And when they coexist, they avoid competition by occupying a slightly different niche. They don't usually occupy the same niche. So these three groups of earthworms give us a really good opportunity to research the evolutionary pressures involved in the evolution of giant body size, but also the molecular mechanisms leading to it and the effect of those really impressive earthworms on their ecosystem. Finally, another study that I find really interesting is the study of the evolution of ecological categories. They were proposed by Marcel Boucher, and they were recently defined by Bottinelli by using quantitative methods, and they are defined by functional and behavioral traits. The three main ones are epigic, endogic, and anethic, and they have also intermediate categories. Those at least those three main categories appear to have evolved independently in different lineages and genera. But one large clade that we can see here in that AHE3 concentrates most of the epigeeks and anethics. Of all the epigeek and anethic species, pretty much all of them are here, which is very interesting. In Dendrobaena, Octotrilus, Octolacean, Lumbricus, Sachelius, Bimastos, Eisenoides, Eisenia, Aporoctodea, and Sclerotica. And future work will allow us to identify genes which are under selection and which are shared between each of the ecological categories. By that I mean, for example, that we can find that epigeek earthworms share several genes under selection, while anethic earthworms share a different set of genes under selection. Well, thanks a lot for your attention. Be aware that earthworms are powerful fellas, as you see here. and I want to thank you, my university, for allowing me to develop my research, and also the Franco-Iberian Earthworm Systematics and Evolution Group, formed by Marta Novo, Thibaut Decaens, Jorge Dominguez, and by myself.